Welcome back. So we were just going to start talking about the juxtaglomerular apparatus. So where the efferent arteriole leaves the glomerular capsule, it's going to come in contact with the distal convoluted tubule. And at that area, the distal convoluted tubule has a group of cells known as the macula densa. These cells are going to detect changes in sodium chloride concentration, so the amount of sodium in your system, which is typically associated with your blood pressure. And if you have low concentrations, then that's going to signal the granular cells to produce renin. And then renin is a hormone that stimulates the production of angiotensin II eventually. So, I mean, it starts that stimulation or that cascade, which is going to increase blood pressure and increase salt uptake. And when sodium moves, chlorine moves as well. So it kind of controls both. In the afferent arterioles, we have these juxtaglomerular cells. These juxtaglomerular cells are going to detect changes in plasma osmolarity. So if you have a high osmolarity, lots of solutes in the plasma, that's going to stimulate these juxtaglomerular cells to constrict. And when they constrict, that's going to then decrease the amount of fluid released. It increases blood pressure and also stimulates the release of renin, which will also increase blood pressure. This is going to increase the amount of fluid in your body and decrease the plasma osmolarity. So if you have low plasma osmolarity, these cells are going to relax and more filtrate can be produced. And so here you can see the macula densa cells right here, part of that distal convoluted tubule. When they get stimulated, they're going to secrete renin. If in the afferent arteriole, the juxtaglomerular cells recognize high levels of osmolarity in the blood plasma, they're going to constrict, and they're going to also send signals to tell the macula densa cells to secrete renin. Eventually, renin will enter the system. When renin enters the blood, renin is going to eventually come in contact with angiotensinogen, which is produced by the liver and released and kind of moves through our system in an inactive form. So when renin and angiotensinogen come together, they form angiotensin I. Angiotensin I, then when it moves through pulmonary circulation, is going to be converted to angiotensin II by angiotensin converting enzyme, or ACE. So angiotensin II is what stimulates strong vasoconstriction of our blood vessels. It stimulates the release of antidiuretic hormone. So it tells the hypothalamus, hey, we need antidiuretic hormone. So we increase the amount of fluid uptake by aquaporins. It's also going to tell the adrenal glands to release aldosterone. And that's going to increase the amount of sodium, which increases the amount of fluids we take in. So we're going to be increasing the amount of fluid in our blood. And that's going to bring our blood plasma osmolarity back to normal. And so this is just your figure, the whole thing. So I was just showing this front part. This is just the whole picture of the juxtaglomerular apparatus. From the glomerular capsule, we move into the renal tubule. The renal tubule has three main parts. We have the proximal convoluted tubule, we have the loop of Henle, and then we have the distal convoluted tubule. The proximal convoluted tubule is the initial portion of the renal tubule, and this is where the majority of reabsorption and secretion occur in the nephron or in the renal tubule. That's what I'll say. The reason for this is they have microvilli in the proximal convoluted tubule, which increase surface area for reabsorption. So from the proximal convoluted tubule, materials move into this loop of Henle. You have a descending loop of Henle and an ascending loop of Henle. The descending loop of Henle has, well, both the descending and ascending loop of Henle have different regions where you have thick and thin epithelium. In the thick regions, you're not going to have as much reabsorption. In the thin regions, you're going to have more reabsorption of materials. But then in the distal convoluted tubule, so here's what I was talking about. So you start out, you have this descending loop of Henle, and then at this hairpin here, you have the ascending loop. And in each side, we have some regions of the descending and some regions of the ascending limb that are going to be thick or thin. At the thin regions, we're going to have a lot of reabsorption and secretion. The thick regions, we're not going to. And then we have this distal convoluted tubule where anything that has to be secreted from the blood is going to move in because this is the last chance it has. So not as much reabsorption or secretion occurs in the distal convoluted tubule, but there is still some, specifically certain drugs and water. And then we end up in the collecting duct. And so for every nephron, or for every collecting duct, there are multiple nephrons that empty into that same collecting duct. The collecting duct is going to then bring the fluids to the papillary duct, which then drains into the minor calyx, then to the major calyx, then to the renal pelvis, then to the ureter and the bladder and the urethra. So this is kind of the drainage from the collecting ducts. This is just how fluid or urine moves away from the body, okay? So we're producing a lot of filtrate. Like I said, again, I've said it multiple times already, but I'll say it again. About 25% of the urine or of the blood in our body at any given time is going to be being filtered by our kidneys during rest. So we produce about 180 liters of filtrate every single day, and hopefully you remember how much blood we have in our body, but I very likely, no, I'm asking, think about it right now. How much blood do we have in our body, and could we sustain losing 180 liters of fluid? And hopefully everybody said, no way, we would be dehydrated and dead. And that is correct. The majority of that filtrate is going to be reabsorbed. So a lot of it gets reabsorbed in the glomerular capsule. And then that doesn't get reabsorbed there is going to get reabsorbed in the proximal convoluted tubule, or the loop of Henle, or the distal convoluted tubule. So by the end, we only have a very small amount of materials actually leaving our blood every single day. Only about anywhere between half a liter to 2 liters of fluid are going to be released in general. And that depends on hydration levels, so it could be a little bit more. And also, I mean, if you have certain diseases, you may produce more liters of fluid than others. So what controls the fluid or the filtration rate? There are two major pressures that are going to control the amount of filtration we produce or filtrate we produce. So we're going to have the hydrostatic pressure and the colloidosmotic pressure. So our blood hydrostatic pressure is the pressure pushing fluids out. The capsular hydrostatic pressure is the pressure pushing fluids back in. So this is just that hydrostatic pressure of the fluids on the walls trying to say, get out, get out, get out, right? And then we have the colloid pressure. Colloidosmotic pressure is what's pulling fluids, so that's those proteins that are saying, hey, you need to stay with the blood. And so we have a higher colloid osmotic pressure in the capillaries, and so that pulls fluids into the blood. So if we take the blood hydrostatic pressure and we subtract the colloid osmotic pressure and the capsular hydrostatic pressure, we get a net filtrate pressure. So this here is the amount of fluid that will actually leave. So if we change any of these numbers, so right here I changed the hydrostatic pressure. I made it higher. So now we have a higher amount of pressure pushing fluids out, but everything else stayed the same. That is going to cause us to have more filtrate. So think of having a higher hydration level. If that pressure decreased, that would cause us to have a lower hydration level, and that would cause us to not produce as much filtrate. So tubular reabsorption and secretion occur along the entire nephron loop, but the majority of reabsorption occurs within the proximal convoluted tubule. So that's the first area of the nephron loop. And that's because it has those microvilli that help to increase surface area for absorption. So we have a lot of reabsorption of materials that were released by that filtration membrane. And there's going to be secretion of materials that couldn't fit through the filtration membrane. So if we move down, you see a lot of water is being reabsorbed, and so water is reabsorbed along the entire line, but we have a lot of water absorbed here at this thin loop of Henle, and then we have some secretion into the blood or into the tubular fluid, and then up here we have some ions being moved out of the loop of Henle. And up here again we have more reabsorption and a little bit of secretion. So secretion occurs throughout the entire loop. In the collecting duct, the only thing that typically is going to be secreted is going to be water. So nothing is reabsorbed in the collecting duct, but you're still going to have, depending on your hydration level, water and possibly urea being secreted out. The urea would help to maintain a high osmotic gradient in the kidneys. So how do these things get reabsorbed and secreted? Through all the different transport mechanisms you learned last semester. So remember active transport, sodium potassium pump, endocytosis. There's also secondary active transport using symporters or antiporters, where you have one molecule moving down its concentration gradient and another molecule moves against its gradient with that molecule. That would be a symporter, or moves in the opposite direction but uses the energy of the molecule moving down its concentration gradient, the ion gradient. That would be an antiporter. There's passive transport like diffusion, facilitative diffusion, and osmosis. Okay, so this table is from your textbook. I'm trying to see where this table is located, but I'm not seeing it right now. I know it's from your textbook. But there it is right there. It's on page 1223 if you're looking for it. This table shows what is reabsorbed at which area, or the amount of material reabsorbed at each area, what is being secreted, and the mechanism by which it occurs. Materials that are being reabsorbed by or in the proximal convoluted tubule are moving into the peritubular capillaries. Anything secreted would also be being secreted by the peritubular capillaries. Surrounding the distal convoluted tubule and the majority of our loops of Henle, the ones on the cortical nephrons, those ones are going to also have peritubular capillaries surrounding them, and so reabsorption occurs into those peritubular capillaries and secretion occurs into those from those peritubular capillaries. Surrounding the juxtamedullary nephron loop of Henle, we have a vasa recta. And so that's going to allow for reabsorption and secretion of materials from the loop of Henle and into the loop of Henle of those long juxtamedullary nephrons. I'm going to stop here because this has been 15 minutes. So I will talk to you guys about those long loops of Henle in the next video. Bye.